wasn't until 1854 that Gregor Mendel has conducted his experimentation to understand how the trait being transmitted from one generation to another. So in this video, we are going to explain how Mendel did his work. Hello everyone, my name is Arseni Aziz and I am the creator of Biology with Arseni, and today we are going to talk about how Gregor Mendel has conducted his experimentation to understand how the trait being transmitted from one generation to another. Gregor Mendel have gone through several steps, have gone through several years to understand how these trait in order to become the developer and the creator of a certain branch of biology that we call it today the genetics. We owe Gregor Mendel that much to understand how he conducted his experimentation and how he performed these experimentation to make it understandable to see how the organism interact with each other and why trait is being transmitted from one generation to another and how. So as you can see over here, this is Gregor Mendel, the father of genetics. Now, Gregor Mendel, he was too many things. He was a monk, he was a person who understand how to form a plant, and he had studied math. Specifically, he had a well knowledge with statistics. So Gregor Mendel in one day had decided to understand how the trait in some organism how it's being transmitted from another. But the idea is, during the understanding of how these traits are being transmitted to another, Gregor Mendel find it hard because it takes time, it takes years to develop the trait and to understand it. So he came to the next best thing. He used the bee plant in his study. And in his study, he used the probabilities to understand how these ratio of this trait is being done. So why the bee plant? Gregor Mendel, based upon his background of having an idea how to use a plant because he was a peasant and he had a knowledge with farming, so he tried to use the bee plant because of so many things. One of the things is because it has a rapid life cycle, it produces lots of seeds and lots of generation, and the characters are observed clearly. Mendel found that there are 7 characters, and these characters, they are, you can say that they have an opposite range of spectrum, okay? Like the seed shape, you can have an either round or wrinkled, like the seed color, you have either green or yellow. You have the flower color, which is a purple or white. You have the pod shape, whether it is inflated or constricted. The pod color, whether it is green or yellow. The stem length, whether it is short or long. And the flower position, whether it is axial or terminal. So Gregor Mendel have decided to test the bee plant for each and every one of these traits to understand these 2 words up here. What is the word dominant and what is the word recessive, and why some plants they appear purple and why some other plants and other flowers they appear white, why some are long, why some are short, and etc. of these traits that Mendel had used. So in order to do that, Mendel had performed the first mating to ensure the purity of the plant for a certain character. For instance, over here we have 2 flowers, okay, 2 bee flowers. One of these flowers which is a purple and one which is white. So he made sure that these 2 traits are pure for several generations. Let's say an 8 or 7 or 8 generation, okay, and then he allowed a purple flower to have a mate with a white flower. And the result for Gregor Mendel that he has the generation for these flowers with a purple flower color. Okay, then he allowed that generation that produced a purple flower to have a self-pollination. So he found that exaggerately something weird for him. He found that the white trait, that is aka recessive for us now, okay, that it appear but in a ratio for every 3 flowers that had a purple color, we have almost 1 flower with a white color. So Gregor Mendel tried to let's say to put his notes in understanding. So he said that one form of the feature, such as the tool or the color, in that case the purple color, always cancel the other form, okay, of that of the other trait. Okay, so he called that the visible form, okay, the one that is visible and the one that is always appear at any time, he called it a dominant, and he called the hidden one, he called it recessive. In the second generation after the plant were self-pollinated, okay, now the hidden form reappeared, okay, in a ratio, specific 3-to-1 ratio. Okay, and Mendel found that the feature was inherited independently. One feature, like the plant height, didn't influence any inheritance of any other feature that Mendel is being tested for. So he tried to put it in some rather fancy way. Okay, so he called that the flower color and the height. They are controlled by a pair of heritable factors that we later have called them alleles. Okay, and one version of that factor is a dominant form that could mask and hide the effect, or the other version, which we call it the recessive form. And these two paired factors separated during the gamete production, okay, whether this gamete is sperm or a pollen grain or an egg, okay, and the other one receive the other factor, okay, and that happened randomly. That don't happen in any kind of, let's say, uh, any kind of organization or any kind of predetermination. And the factor that controlling these different characteristics, they are inherited independently of one another. So why this pattern? Okay, so Mendel have put two laws that explain why this pattern. First one is the law of segregation, and according to Mendel, law of segregation, that only one of the two gene copy present in an organism, and it is distributed to each gamete that it makes that it makes, and the allocation of the gene copies is random. That means you get one copy of the trait or one copy of the character, and that gamete that get this character, it's completely random. It don't happen by predetermination, like you are going to get that and the other one get the other form. No, that happened in a very random case and in a very uncontrolled manner. Okay, so Gregor Mendel found that if, in order to prove himself right, he brought, let's say, a purple flower, okay, and he made it sure that it's pure, and it is called a homozygous because it has two exact copy of the same character. Okay, so he got one dominant uh, uh, parent, okay, for a certain character, and he made sure that is pure, and other parent that is recessive for a certain of that trait. Okay, then the gamete formed, one gamete get one copy of that, and it produces 100% dominant character, according to the law of dominance, that the dominant character always mask the other trait and always mask the other allele. Okay, so it forms a 100% dominant character, but it has an heterozygous formation or a hybrid one. So if he allowed that these two heterozygous to self-pollinate, and by the word self-pollinate, it means that the pollen can move from the same flower to the female reproductive organ on the same flower. Okay, so it forms uh, the self-pollination like that. And in order to understand it, Mendel has used the Punnett square in order to explain the probability, and he found out I get a ratio of 3-to-1, 3 of the dominant character to 1 of that recessive character, okay? And the probability helped him in order to explain how this happened and in order to understand how these ratio happen. Because there were too many people before Gregor Mendel, but they didn't integrate math in their explanation, and they didn't get math say how did exactly happen and how everything did exactly appears. Okay, so That led to another experimentation by Gregor Mendel that we call it the test cross. Okay, and the idea of the test cross that I can identify the purity of an individual, whether is that individual is completely homozygous or completely pure individual, or it is a hybrid. So in that case, if I have a red flower and the red color over here is the dominant form, and I have another flower which is a yellow flower, and the yellow color is the recessive form. Okay, and there is a note that is so important for you all to understand, that always the recessive is a homozygous or is pure, because in order to show its trait, it need always to be in the pure form. It can show its trait when it is in a hybrid form or any other case. It need to be always pure. So I want to know, is that red flower, is it a pure for the red color, or it is a hybrid? So Mendel had put two characteristics, and that is the use of the Punnett square. So in case if I'm having a pure individual, I'm going to have a 100% dominant character appear. In case of a hybrid, I will have a 50% of that generation over here, like I'm using this Punnett square. Okay, I have an R capital, R small, versus the R small, R small. So I'm going to have an R capital, R small, R capital, R small. That 50%, and the other one is R small, and R small, R small. That is another 50% for the recessive trait. So I have a 50-50 ratio, or 1-to-1 ratio. If we have a 1-to-1 ratio, that means we are testing a hybrid. If we are having a 100% dominant character, that means I'm testing a homozygous dominant form. Okay. The other law that Mendel had developed is the law of independent assortment, because there was an idea, and that idea led to a center understanding. For an example, in human, um, people have always said that, let's say, if you're having a if you ever having a blue eyes, you'll always have a blonde hair, okay, or you're going to have a white skin. So that was not the case. Okay, and that wasn't the idea of that. So Gregor Mendel law of independent assortment have explained that the alleles, okay, they are sorted into independently. They are distributed in the gamete independently from one another. I mean, if I have, let's say, two or three traits, these two or three traits I'm talking about, they are sorted independently. They are distributed independently from one another. We don't have any kind of, let's say, predetermination that you're going to have 1 and 2 and 3 together, okay, and you're going to have 4 and 5 and 6 together. No, that completely happened in random case for each allele and for each gamete. So you don't have predetermined um, gamete formation, or you don't have predetermined allele formation in your gene. So, in order to understand that, Mendel had formed the idea of a dihybrid cross. The idea of a dihybrid cross is very simple. You are making a hybrid for not only one trait, you're making a hybrid for two traits. So, in order to form that hybrid, you need to make sure that you are getting a one individual that is pure for two traits, not only one trait. You want to have an individual that is pure dominant for the two trait, and another individual that is dominant for that is recessive for the other two trait. So for example, we are going to test here the round and the wrinkle bees. So we are giving the round R capital, and we are giving the wrinkle R small, and we are going to give it for the length. So the L capital is for long stem bees, and the L small for short stem bees. So in the first one, Mendel performed a normal mating for an individual with R capital, R capital, L capital, L capital, which is a dominant for both trait and pure for both trait, and R small, R small, L small, L small, which is also recessive for these two traits. So the generation that is produced from here is R capital, R small, L capital, L small. By this, I'm having a different combination. For gamete. When a gamete is formed from that individual, I have 4 probabilities for it. Okay, imagine that we are having 2 individual with that formation. Okay, that will bring us to a 16 probability to search about it. Okay, now these 16 probability, if you can see it over here, out a little bit so you can see clearer. So I'm having an individual with an R capital R small, L capital L small, mating with another individual with an R capital R small, L capital L small. So you are getting 16 probability. You have 4 type of gamete that could form from one parent and 4 type of gamete that could form from another parent. So I'm having here a 16 probability. So here we are having 16 probability. So having a 16 probability over here, that brings us to how these form. So I have the first one over here. So I have an R capital, L capital, capital. So this is a pure dominant. And I have at the end over here R small, L small, L small, which is a pure recessive. Okay. So if we are continuing the formation of these matings, so I'm going to have the following mating that shape. So I'm mating this with the first one with the second one over here. So I'm getting an R capital, L small, then R capital, R small, L capital, and the other one R capital, R small, L capital, L small, and then R capital, L capital, L small, and it goes on and on. This 16 probability ratio of a 9, 3, 3 to 1 ratio. That ratio is so important. Okay, now that 9 to 3 to 3 to 1 ratio, it shows the ratio of traits that appear in the second generation. Of course, it appears in larger numbers, but when you try to get a ratio for this number, you get the 9:3:1 ratio between 1. I have 9 with a round long stem. I have 3 with a round short stem, and have 3 with a wrinkled long stem, and I have 1 wrinkled short stem. And that brings us to another thing. Okay, now these 9:3:1, it shows a certain thing, and that thing, it shows that round long stem or round short stem or wrinkle long stem or wrinkle short stem, it shows how the organism appear. So how an organism appears, the thing that you notice, okay, that we call it a phenotype. Okay, now how the organism appear for you, how you see that organism, this is the, let's say, it is the trait that you can see in, is the physical trait that you see in front of your eyes. You see it, let's say a purple, you see blue, you see green, you see it yellow, whatever the color you see it, it's long, it's hair, it is the physical trait that you can see. But over here in the table, you can see that there is something different. Okay, you see we are here talking about an R capital, L capital, L small. We are talking about the formation of these alleles. So the formation of these alleles from the inside, we here are talking about something that we don't see. We are talking about the formation of the trait itself, okay? And that is called the genotype. It is the genetic content and how this genotype is formed. It is what is inside and how this trait appear for you. It is the phenotype, but from the inside, this is the genotype. This is the genetic content of it. This is how the allele and how the trait is formed from the inside. Okay, so the law of independent assortment proved that these traits are inherited separately from one another. It is not necessarily that you will find a trait that is always accompanied by another trait. Okay, that is for another case, and that's for another episode that we are going to talk about. Okay, so what Mendel did was a breakthrough for all of us to understand how the genes and how the traits are actually being proven and how these traits are being developed. So thank you all for today, and I hope you like our episode. If you have reached to that point, don't forget to give us a like, subscribe, and always activate the bell ring that down here, okay? So you can reach and you can have any notification that tells you that we can have made another episode, so you can watch it. And of course, don't forget to share this episode so you can share it with your friend and let the knowledge pass to everyone. So if you have any comments, if you have any question, feel free to comment below. I respond always fast. So get always share, always like, always subscribe, share it with your friend. Until I see you next time. I was Arsene Aziz, and that was Biology with Arthane. Goodbye, everyone.